Good evening, all of you. So today we are going to discuss specifically about the role of air in photosynthesis. So how air influences photosynthesis, air and photosynthesis. So for this purpose, we have to discuss about a great scientist who is named as Joseph Priestley. Joseph Priestley. So why exactly we are giving importance to this person? He has performed several experiments where he has shown that oxygen is liberated from plants, and that oxygen which is liberated from plants is consumed by the animals like us. See, animals like every organism which is living upon the earth consumes this oxygen which gets liberated by plants. So that was proven by Joseph Priestley by a famous experiment called bell jar experiment. So I told you, Joseph Priestley has discovered oxygen. And who named the oxygen? So, who coined the term oxygen? There is one more scientist called Lavoisier. Lavoisier is a scientist who has named oxygen, and Priestley is the scientist who discovered oxygen. So, we are now going to discuss this particular activity in order to know the importance of the gaseous exchange which takes place between the animals and plants. So in a bell jar experiment, we need mice, bell jar. I'll explain what is bell jar. Bell jar, mice, a candle. Along with that, we need a mint plant. These are all we need to perform this activity. This activity was performed by Joseph Priestley. So, see what exactly he has done using this particular apparatus. So, we shall see bell jar is nothing but a jar which is in the shape of a bell. You know, the bell shape is like this. A bell shape would be like this, right? So, he has used exactly a jar which is in the shape of a bell. That is why it is called as a bell jar. So, what exactly he has done using this bell jar? You see here, in one setup, he has placed, see, this would be sealed, okay? This bell jar is placed upon a platform. If you consider a platform like this, a bell jar is placed upon a platform like a floor or something like that, and the walls of this bell jar would be seized. See, the connection which it is making with the surface will be sealed. See here, if the bell jar is placed upon a platform, then obviously this particular part would be sealed. This particular part would be sealed to the platform such that no air enters this platform. Here, no air must be entered. Air enter our such that you can ensure that whatever the situation, whatever the condition, whatever the results which we are going to interpret from this must not be a false positive. And the meaning ఏంటంటే పక్కాగా దీని లోపల ఉన్న గ్యాసెస్ వల్లనే ఈ యాక్టివిటీ జరిగింది అని చెప్పడానికి ఇది సీజ్ చేస్తున్నాం. బయట నుంచి గాలి వస్తే ఏంటి, లోపల ఉన్న గాలితో మనం చేసిన యాక్టివిటీని ప్రూవ్ చేయలేము. You have to ensure that nothing enters this particular bell jar. Before placing this bell jar, he has placed a candle. And what he did, he has lit this candle. He has lit this candle. So then he was able to see, after some time, he started observing this candle. After some time, the candle was put off. The candle was put off. ఎందుకు పోయింది? Because Yeah, he didn't say anything as such at that time. Let us keep it aside, and later we shall see what happened to this particular candle. Now again he has taken one more equipment, and here what he did was he has taken a candle, and along with this candle he has assumed a rat under this bell jar. So now you see here, after some time, this mice got suffocated and it was dead, or else it has become unconscious. And later, this candle was also put off. So both these mice and this candle was put off, as there is no availability of something which keeps them alive. ఏదైతే వాటిని alive ఉంచుతుందో అది దాని లోపల దొరకట్లేదు. నైస్ alive ఉండేది, బయట క్యాండిల్ గ్లోయింగ్ కండిషన్ లో ఉండేది. బట్ ఈ బెల్జార్ కింద పెట్టేసరికి ఇట్ వాస్ పుట్ ఆఫ్ అండ్ mice got, what we say, suffocated. Why this has happened? There is something which is keeping them alive outside. ఈ బెల్జార్ బయట వాటిని ఏదో alive గా ఉంచుతుంది అని చెప్పేసి Joseph Priestley has a thought. Then he started assuming what is a plant is kept in that bell jar along with this candle and mice. So what he has did was he has kept this candle and a mice along with the candle and a mint plant. A mint plant was kept along with these, what we say, candle and mice. So here also see same thing happens, everything must be seized. So what exactly happens here? He has lighted this candle from outside using the concept of converging the rays. Okay, magnifying glass తీసుకొని he converts the sunlight upon this candle and he has lit from outside. Now we can use a modern technique. ఇదే యాక్టివిటీ ఇప్పుడు మనం చేస్తే we can use electric spark, we can use some, what we say, magnifying glasses similar to focus lights ఉంటాయి. ఇప్పుడు highly focused lights ఉంటాయి, ఆ focus lights తో లైట్ చేయొచ్చు. So by converging the rays upon to the candle, so బయట నుంచి ఈ candle ని లైట్ చేసింది. So this one and the mice both are surviving, but whereas here they got put off and the mice got suffocated. As Rishikesh has asked, plants are the only source of oxygen. We can say that the plants are the only source for the oxygen, really, because they are the only beings which can fix CO2 and liberate out O2 by using a renewable resource of energy. We cannot produce oxygen with a renewable source of energy. The plant has special ability, it can fix CO2, it can use up water and liberate out O2 by using sunlight. So that is the reason the plants are to be planted in order to ensure that this entire planet Earth, the living organisms which are on the planet Earth, are survived. So here you see the candle is lightning and this mice got it's like mice is also living, whereas in this case mice got suffocated, light was put off, here also light was put off. What exactly happening was there is a gaseous exchange which is happening between the candle and the mice. So what exactly is happening? This candle is liberating out some gas and this mice is also liberating some gas. So those gases which are liberated out of these candle and mice are being used by the mint plant, and mint plant is also liberating some gases which are useful for the burning of candle and the life of mice. So here he has a thought that there is some gas which is being liberated by the, what we say, mice and candle, and which is used up by the plant, and there is a gas which is being liberated by the mint plant, such that there is a gaseous exchange taking place. Here that gas which is being given to this candle and mice was oxygen, and that was named by Lavoisier. So now you see, we shall see if you go for an activity. So from here you can say that the CO2 is used by the plant, and what we say, O2 will be liberated by the plant. If we go to the reaction which was given by C.B. Van Neel or else Robert Hill, whatever is the reaction, once we shall see the reaction which was given by C.B. Van Neel, that is CO2 plus 2H2O, which liberates CH2O plus H2O plus O2. This was given by C.B. Van Neel, if you remember in my second lecture. If you remember my second lecture, in the second lecture we have discussed C.B. Van Neel reaction. This is C.B. Van Neel reaction. From this reaction, if you are able to observe, the plant uses CO2 and plant liberates of O2. So here we can say that O2 is being liberated and it is used up by the organisms. That was proved by Joseph Priestley in a Bell-jar experiment. Whereas we have to see whether exactly CO2 is used by the plant or not through an activity. This activity is named as Mohl's half-leaf experiment, or else an activity to show CO2 is used by the plants. So Mohl's half-leaf experiment, as this experiment was conducted by Mohl, so it is called as Mohl's half-leaf experiment. Mohl's half-leaf experiment. So we shall see what exactly is happening in this Mohl's half-leaf experiment. What are the apparatus first we have to we require to conduct this particular activity. So what are the apparatus required? First one, you have to take a plant such that its leaf is a bit lengthy, like Ashoka tree's leaf. Okay, lengthy leaf, not like banana. It must be narrow and lengthy. It must be narrow and lengthy. Banana leaf good inta pedda untadani banana leaf తీసుకొని activity చేస్తా అంటే it's not going to work. You have to take a narrow leaf and which is a lengthy one. Okay. So you have to consider a plant which has a narrow and lengthy leaf. Narrow and lengthy leaf. Next, a plant with narrow and lengthy leaf. Second thing, you have to have iodine or betadine. In the activity, in the lecture number 3, we have discussed starch presence test. That test is very, very important in order to understand these activities even. We need KOH, potassium hydroxide. Potassium hydroxide. See, potassium hydroxide exists as pellet as sodium hydroxide. So many of you have seen sodium hydroxide. Similar to sodium hydroxide, potassium hydroxide will also have some pellets. It will exist as pellets. Chinna tablets laga untayi, chinna pellets laga untayi. So these pellets are used in the bottle. So instead of solution, you can use pellets, or else you can consider solution also. Both you can consider solution, potassium hydroxide solution, or pellets. See, knowing the apparatus required is very, very important. Okay, so next, fourth one, a bottle which has narrow mouth, okay, which can fit this leaf into. Leaf ni din lopaliki పెట్టేటట్టు ఒక చిన్న మౌత్ ఉన్న బాటిల్ తీసుకోవాలి, a bottle. And fifth one, a rubber cork with split. Rubber cork which is splitted. splitted rubber cork, okay, which is splitted rubber cork ante, see, if there is something like this, there must be a split like this, chinna split undali nada. Splitting ante oka chinna opening, itla small opening, narrow opening undali, rubber cork lopala opening undali, dan lopaliki manam leaf ni pettachu. So we shall see what happens with the activity. You need a plant which has narrow lengthy leaf, iodine, KOH, bottle, rubber, I mean, yeah. So all this you have to consider. Now I am going to tell you a specific process of destarching. Destarching etla chestamo chudam. We shall see about destarching a plant. So what exactly is destarching? Destarching అంటే D means something which is removing. Science లో ఎప్పుడైనా D అన్న ఒక శబ్దం వస్తాయి. Whenever you experience a sound called D, DE, dehydration, destarching, D. Yeah, I mean I'm unable to recollect some words. Destarching or dehydration, whatever it is. D means remove. Okay, D means removing. So what do you mean by starching? Removing the starch. Removing the starch. So how exactly you can remove the starch from the leaf? First of all, you have to remove starch from the leaf. దీనికి ఏం చేయాలా, దాన్ని ఫర్దర్ గా ఫోటోసింథసిస్ చేయకుండా చేయాలి. The plant, you have to ensure the plant further not perform this photosynthesis process. ఎట్లా ensure starching process and ఏంటి? You have to keep plant in a dark place for 2 to 3 days. 2 నుంచి 3 days dark place lo you have to place this plant. When you place this plant in a dark place, what happens? It cannot perform photosynthesis such that whatever the available starch will be used up by the plant for the survival. అంటే అప్పటి వరకు ప్రిపేర్ చేసుకున్న స్టార్చ్ ప్లాంట్ లోపల ఉంటుంది కదా, లీవ్స్ లోపల స్టార్చ్ ప్రిపేర్ అయి ఉంటుంది. ఇప్పటి వరకు ఫోటోసింథసిస్ చేసి స్టార్చ్ ప్రిపేర్ అయి ఉంటే ఆ స్టార్చ్ ని the plant will start using because there is no further food production within the plant. Same మనలాగానే మన హ్యూమన్ బీయింగ్స్ ఏం చేస్తాం, ఎప్పుడైనా అన్నం తినలేదు అనుకో చచ్చిపోతామా, ఒక పూటకి టూ ఐ మీన్ 1 day చచ్చిపోతామా మనం అన్నం తినకపోతే? నో, నథింగ్ హ్యాపెన్స్ టు us because there will be storage which will be within our body that will be used up. మన బాడీలో ఉన్న లిపిడ్స్, ఫ్యాట్స్ ని యూజ్ చేసేసుకుంటారు మన బాడీ, అంటే అవైలబుల్ ఫుడ్ కంటెంట్ ని వాడేసుకుంటుంది. సేమ్ థింగ్ హ్యాపెన్స్ విత్ ద ప్లాంట్. ప్లాంట్ అప్పటి వరకు ఫోటోసింథసిస్ చేసి స్టోర్ చేసిన స్టార్చ్ కంటెంట్ ఉంటుంది కదా, దాట్ స్టార్చ్ కంటెంట్ విల్ బి యూజ్డ్ అప్ బై ద ప్లాంట్ ఫర్ ఇట్స్ సర్వైవల్. అది బతకడం కోసం అప్పటి వరకు ఉన్న స్టార్చ్ కంటెంట్ మొత్తం యూజ్ చేసుకుంటారు. ఒక 2 to 3 days వరకు ఇఫ్ యు ఆర్ స్టాపింగ్ ద ప్లాంట్ ఫ్రమ్ ఫోటోసింథసిస్, పర్ఫార్మింగ్ ఫోటోసింథసిస్, then the starch will be used up. So you can say that the starch is removed from the leaves. Leaves నుంచి మొత్తం స్టార్చ్ యూజ్ అయిపోతుంది కదా, ప్లాంట్ యూజ్ చేసేసుకుంటుంది దాని ఎనర్జీ కోసం. So you have destarched the plant by putting the plant for 2 to 3 days in a dark room. Now we shall perform the activity చేద్దాం. ఇప్పుడు destarching అయిపోయింది కదా, ఫస్ట్ destarching చేయాలి. destarching ఎందుకు చేయాలా లాస్ట్ చెప్తా. ఈ యాక్టివిటీ మొత్తం కంప్లీట్ అయినాక ఫస్ట్ destarching ఎందుకు చేస్తాం అనేది మీకు అర్థమయ్యే విధంగా I will tell you. Now we shall see the activity. So okay, sorry. A potted plant. As assumed, you have taken a potted plant which has a lengthy half leaf. Lengthy leaf. Narrow and lengthy leaf. So to this, what I have did was I have taken a bottle. Sorry. So here what I did was I have taken a bottle. In that bottle, I have put a cork. మనం ఒక splitted cork తీసుకున్నాం కదా. I have put that cork into the bottle's mouth. And that cork has a small slit. ఒక కొంచెం పొడవుగా ఓపెనింగ్ ఉంది. ఆ ఓపెనింగ్ లోపలికి ఏం చేసిన I have put the plant leaf into it. Plant leaf split locally I have inserted. Now, I mean, sorry, I forgot. In this, what we say? In this particular bottle, you have to assume KOH solution. You have to take KOH solution in this, or else you can take KOH pellets also. మనం ఏం చేసాం KOH solution తీసుకున్నాం. So KOH solution has to be taken such that this leaf must not touch the KOH solution. Remember that leaf must not touch the KOH solution. So the KOH solution is taken. So KOH solution ఎందుకు తీసుకున్నాము, ఈ యాక్టివిటీ అంటే ఎందుకు చేస్తున్నాము, CO2 is consumed by the plant అని ప్రూవ్ చేయడానికి. ఫోటోసింథసిస్ చేయాలి, స్టార్చ్ ప్రొడక్షన్ అవ్వాలి అంటే CO2 ని కన్జ్యూమ్ చేయాలి అని ప్రూవ్ చేయడానికి this activity we are performing. So what are we doing? We have taken this KOH solution. What is the specialty of this KOH solution? This KOH solution will absorb CO2. This KOH solution will absorb CO2 which is present within this bottle. In the transparent bottle there are sunlight. See, if you assume there is a sun over here, సన్లైట్ లో చేస్తున్నాం మన ఈ యాక్టివిటీ. ప్రీవియస్ గా డీస్టార్చింగ్ చేసేసి సన్లైట్ లో తీసుకొని వచ్చినాం దీన్ని. సో నౌ ద లైట్ ఈస్ ఫాలింగ్ అపాన్ ఎవ్రీవేర్. దీని మీద కూడా లైట్ పడుతుంది. సో వీఆర్ నాట్ స్టాపింగ్ లైట్ టు ఫాల్ అపాన్ దిస్ పర్టికులర్ లీఫ్. లైట్ పడుతుంది, బట్ యు ఆర్ ఎన్ష్యూరింగ్ దాట్ ఇక్కడ నుంచి తీసుకున్న ద వాటర్ విచ్ ఎవర్ ఈస్ టేకెన్ బై ద రూట్స్ విల్ బి పాస్డ్ టు లీఫ్. తీసుకున్న వాటర్ పాస్ అవుతుంది, సన్లైట్ వస్తుంది, బట్ వీఆర్ ఎన్ష్యూరింగ్ దట్ no CO2 will be present. CO2 ఉండదు because we have taken KOH solution. KOH solution ఏం చేస్తుంది, available CO2 తోని రియాక్ట్ అవుతుంది. This KOH will absorb this CO2, means it will react with CO2 such that no CO2 will be available for the leaf. ఈ బాటిల్ లోపల ఉన్న లీఫ్ కి CO2 అవైలబిలిటీ లో ఉండదు, అంటే దీనికి CO2 లేదు. Now you keep it for some few hours, like 4 to 5 hours. 4 to 5 hours. Ensure that there is nothing, no gas would going. ఇక్కడ స్లిట్ ఉంది కదా, స్లిట్ దగ్గర వాసలైన్ పెట్టేసేయండి నాన్న. The slit must be sealed with the vaseline or grease, whatever, because the gas must not enter the bottle. So that is the reason the slit will be sealed. Okay, slit seal అయిపోవాలి, లోపలికి గ్యాస్ వెళ్ళకుండా. ఎందుకంటే ఇక్కడ నుంచి గ్యాస్ వెళ్తా ఉంటే ఇక్కడ CO2 అవైలబిలిటీ లో ఉంటుంది. So we must ensure that there is no CO2 within the bottle. Now this happens after 4 to 5 hours. What I will do is I will take out this leaf out of the bottle. ఈ బాటిల్ లో ఏం చేద్దాం, లీఫ్ ని బయటకు తీసేద్దాం. We shall take out the leaf, everything, and we shall pluck it off from the plant నుంచి కట్ చేసేద్దాం. We shall cut down the leaf from plant. So now the leaf is available. So this part was kept in a bottle. From here to here, no oxygen was, no CO2 was available. But this part CO2 was available. ఇక్కడ CO2 availability లో లేదు, ఇక్కడ CO2 availability లో ఉంది ఈ part లో. So this part was available. Now what I am doing is I am taking this particular leaf and I am performing starch presence test. See, starch presence test was discussed in the previous class, that is lecture number 3 of nutrition, the food supplying system. You can type nutrition food supplying system 3. You will get it. Starch presence test. We will perform starch presence test for this leaf by putting iodine upon it. Iodine. If you put iodine upon this entire leaf, this particular side will turn into blue, dark bluish color, blue black color, but there is no change of color. No change of color was happened in this part of the leaf. E part of the leaf lo em change ledu, no change of color. Why this part was, there is no, why there is no change in this part of the leaf? Because no starch was prepared. No starch was prepared. అయితే ఫస్ట్ నేను చెప్పిన వై వీ హ్యావ్ ది డీస్టార్చింగ్ అని. డీస్టార్చింగ్ ఎందుకు చేసినామో ఇప్పుడు చెప్తా అని చెప్పిన. ఎట్ ద ఎండ్ ఆఫ్ దిస్ యాక్టివిటీ ఐ టోల్డ్ యూ దట్ ఐ విల్ ఎక్స్ప్లెయిన్ వై వీ హ్యావ్ డీస్టార్చ్డ్ ద ప్లాంట్. ఒకవేళ డీస్టార్చ్ చేయలేదు, ఇఫ్ వీ హావెంట్ డిడ్ డీస్టార్చింగ్ స్టెప్ బిఫోర్ దిస్ ఎక్స్పెరిమెంట్, డీస్టార్చింగ్ లేకపోతే ఏంటి, మొత్తం బ్లూ కలర్ గా కన్వర్ట్ అయిపోతుంది. బికాస్ ఎంటైర్ లీఫ్ కన్సిస్ట్ ఆఫ్ స్టార్చ్. లీఫ్ మొత్తంలో స్టార్చ్ ఉంటుంది, అంతమంది ప్రిపేర్ చేసిన స్టార్చ్ అవైలబిలిటీలో ఉంటుంది. దెన్ ఇట్ విల్ ఆల్సో టర్న్ ఇంటూ బ్లూ. So you cannot prove that CO2 must be available to perform photosynthesis. If you destarch it, then you can differentiate ekkada starch form ayindi, ekkada starch form avvaladu, ekkada starch form avvaladu, ekkada starch form ayindi. This particular portion of the leaf was kept in బాటిల్ లోపల మనం ఈ పర్టికులర్ లీఫ్ పార్ట్ ని బాటిల్ లోపల పెట్టేస్తాం. అక్కడ దేర్ ఈస్ నో అవైలబిలిటీ ఆఫ్ CO2 అవైలబిలిటీ లేదు. ఎందుకు CO2 అవైలబిలిటీ లేదు? బికాస్ we have put KOH solution in the bottle. That KOH solution will absorb CO2, which makes unavailability of CO2 for this particular part of the leaf. And we can now say that CO2 is necessary to perform photosynthesis. If CO2 is not available, no starch was prepared. If no starch was prepared, that indicates that no photosynthesis was performed. So this is how the photosynthesis is linked with air. So with this I conclude air and photosynthesis. Next we shall discuss light and photosynthesis. How sunlight is involved in the process of photosynthesis. That's all for today. Bye bye.